I'm Erica Sanders with the Collaborative on Health and the Environment. I want to welcome you to today's CHE partnership call, which is titled EDCs, Oil and Gas Development, and Groundwater with Dr. Susan Nagel. This call is being hosted by the CHE EDC Strategy Group, and our moderator today is Cheryl Patton, Director of Commonwealth's Biomonitoring Resource Center. Dr. Nagel has provided slides to accompany her presentation today. A link to those slides was emailed yesterday to everyone who had already RSVP'd. But if you still need to download them, you can go to CHE's website at healthandenvironment.org and select today's call from the top of the left-hand box. You'll then see the link to the slides posted above the call description. Due to the limited time of this call, we will be taking questions during the Q&A period from the blog only. The link to the blog was also emailed yesterday to everyone who RSVP'd, but if you still need to access it, you can go again to the CHE homepage at healthandenvironment.org and select today's call from that upper left-hand box. You'll then see the link to the Q&A comments page. We will get to as many questions and comments posted there as we can during the Q&A period after Dr. Nagel's presentation. Everyone on the call right now has been muted with the exception of our speaker and our moderator. This call is scheduled to last for 30 minutes, and it is being recorded. And with that, I'm going to turn things over to Cheryl.
Okay. Thanks very much, Erica, for starting the call, and thank you, everybody, for joining in to this very exciting call. I'm pleased and honored to introduce Dr. Susan Nagel, who is Associate Professor at the Obstetrics, Gynecology, and Women's Health Department at the University of Missouri. Dr. Nagel has been conducting some extremely important and innovative research looking at hormonal activity in groundwater, primarily around natural oil and gas production sites, and comparing her data with water looked at from control sites far away from such activities. And what she's found is very disturbing and very interesting, and she's moved ahead and connected hypothetically what the results of possibly some of those results might be in terms of health effects of people who are living in surrounding areas and drinking that water. So without further ado, I want to introduce Susan. Great presentation today, and we're all very much looking forward to it, and thanks for joining us.
Thank you. Can you hear me okay?
Yeah.
Okay, great. So thanks for joining in today. I have a lot of slides. A lot of them are background, and I think a lot of you are very familiar with parts of this topic. So I'm going to go through a lot of the background pretty quickly, and then if you have questions afterwards, please feel free to ask. So the Endocrine Society defines an endocrine-disrupting chemical as a chemical or mixture that interferes with any aspect of hormone action. And so this is different from some of the other definitions, for example, the EPA, which defines an endocrine-disrupting chemical as one that has been shown to do this in animals. So I use this Endocrine Society definition. EDCs are found in many, many products, from industrial chemicals, which we'll talk about today, but also natural products and pesticides and herbicides. Not surprisingly, pesticides and herbicides were designed to disrupt the hormone systems of plants and animals. So we have a lot of conserved receptor systems. It is not surprising that these can disrupt our hormone systems as well. EDCs disrupt hormone action through two main ways, either by directly interfering with that hormone receptor interaction, so kind of the lock and key analogy. They can either bind to the receptor and activate it inappropriately, or they can bind and keep the natural hormone from activating the receptor appropriately. The other major way that EDCs work is to alter the amount of the natural hormone in the body. So I'll talk a little bit today about several different hormones. They're largely in the steroid and nuclear receptor superfamily. They are what we call ligand-activated transcription factors. And really what that means is, you know what, I'm sorry, I did not tell you when I was advancing my slides, which I was told to do. I am on slide number 5. So, pretty simply, estrogen receptor, for example, when estradiol, the natural hormone, binds to that receptor, it can turn on gene transcription. And so what that means is it controls all of the ways that estrogen-responsive genes are turned on or off. So if a exogenous hormone, a chemical that was not made by the body, can also do this, it can do it in an inappropriate manner. So estrogens are very small lipophilic molecules. That means that they don't really like to dissolve in water. They prefer to associate with fat molecules. So in blood, they are associated with serum-binding proteins like sex hormone-binding globulin and albumin. And it's only this small fraction shown in slide 6 that is able to diffuse in to cells and bind to those nuclear receptors. And one of the common endpoints of this action is stimulation of cell proliferation, slide 7. So on to slide 8. Why is endocrine disruption important to human health and disease? And in short, it's because hormones and endocrine disrupting chemicals can act at very low concentrations. Therefore, since humans are exposed to many of these chemicals at rather large concentrations, the exposure can be within the range of biological activity. The other major factor is that developmental exposure can alter adult health and disease. So in slide 9, as we know, hormones are essential for normal health and development. And this is kind of an extreme example, but nonetheless absolutely accurate, that it is the presence of fetal testosterone that resulted in sexual differentiation of Brad, and it's the absence of fetal testosterone that resulted in sexual differentiation of Angelina. So while those are extreme examples of the presence or the absence of fetal testosterone, you can see the exquisite importance of these steroid hormones during development. Slide 10 summarizes a huge body of research on fetal and early life exposures to endocrine-disrupting chemicals and their association with adult disease. A lot of what we know is from laboratory studies, where we can make those sorts of cause and effect relationships. And so summarized there are just a number of disease and altered health endpoints that have been associated with either exposure to exogenous chemicals like pollutants or endocrine-disrupting chemicals, altered nutrition, and also maternal health and disease can dramatically alter the fetal environment and program the fetus and result in increased risk of disease in adulthood. So with that very brief intro into EDCs today, we'll move on to our unconventional oil and natural gas extraction as a potential source of endocrine-disrupting chemicals. So slide 12 summarizes that process of unconventional oil and gas extraction, which is the combination of hydraulic fracturing, where we are injecting millions of gallons of water under high pressure with thousands of gallons of chemicals, with horizontal drilling. Slide 13 shows the fracturing fluid composition. It is largely water. The 1% that is not water is made up of proppants, so things to hold open those fractures underground, and then about 0.5%, which is a very, very rough estimate, is chemicals. Now, while that is a small fraction, it actually is thousands of gallons of chemicals per well that is fracked. And each well pad has many wells on it and is fracked many times. So I first became interested in this topic when the folks at the Endocrine Disruption Exchange described that of the 350 chemicals that they had information on used in the process, over 37% had some indication that they were endocrine-disrupting chemicals. Now, the problem with hydraulic fracturing, or that really represents a challenge, is this millions of gallons of wastewater that's produced in the process. So flowback returns immediately to the surface, produced water returns over the life of the well, And this wastewater contains the chemicals that were injected down, but it also contains chemicals that are naturally occurring that are liberated from far underground. So it's this wastewater, the spills and leaks, disposal of, that we think results in contamination of water. So our hypothesis was that surface and groundwater at natural gas drilling sites, spill sites, would contain more endocrine-disrupting activity than reference sites. And note I said activity. In this first study, we did not measure the chemical composition. So in slide 16 shows a map of Garfield County, and inside the box is our sites that had some sort of spill or accident or preventable discharge related to wastewater and unconventional oil and gas processes. Outside of the box shows reference sites that we also sampled that did not have a spill and had relatively few drilling activities around it. So simply what we did, or I should say not simply, but a summary of what we did in slide 17, we collected water in 1-liter bottles, we took it back to the lab, and we used actually glass solid phase extraction cartridges to concentrate the chemicals. We concentrated them and ultimately used tissue culture cells. And what we ended up testing was concentrated 4 and 40-fold over the original water concentration. So just to be clear, that what we were testing was not the water itself, but a 4 or 40x concentration of that original water. And we used a reporter gene assay system, so whole cells, so human cells that had been transfected with reporter genes for different receptors of interest. So what we found in the first study in slide 18 was 2 to 3-fold increased activity in those spill sites compared to our reference sites. And while you see in groundwater there, we had a little bit of anti-androgenic activity at all the sites, we didn't have that at all in our reference sites. We also had elevated estrogenic activity. The next slide shows surface water, which of course is much more complex. However, we did see the same trend that we saw anti-androgenic activity in all of those sites in the Colorado River that drains this entire area, except for one site, Site 2. And we did not see this in the reference site. So with this kind of initial study, we became much more interested in this topic. We found an association, and we want to move forward to test that association for causation. We were involved in a collaboration with the USGS that looked at a disposal facility in West Virginia. So this was an injection well that was used to dispose of unconventional oil and gas wastewater. So this site was particularly amenable for us because it had a creek that flowed from above the site, through the site, and then down below the site. So as you can see in slide 20, as a map, an aerial of number that what's labeled as 4 as the background is above this site. So now we moved on to look at an additional type of hormone endocrine disrupting activity in this study. So in slide 21, you can see that the blank, so these are just our controls, and then the background didn't really differ in endocrine disrupting activity. Then when we got a little bit into the site, number 6, we saw increase in hormone disrupting activity. But what we really saw was at sample sites 7 and 3, which were just a little bit downstream of this injection site, and then a little bit further downstream, was enormous activity. And so what I showed you in the previous slides from Colorado was largely from water that had been concentrated 40x. Those were the results I showed, even though we tested 4x and 40. What I'm showing you here is 4x. So this is 10-fold more diluted than the previous slide. So all in all, what this means is there was enormous endocrine disrupting activity downstream of this injection well pad. So our hypothesis then is that chemicals used in hydraulic fracturing will directly disrupt hormone receptors. And so what we have now done is extend this analysis to 5 different receptor types, shown in slide 22. So we tested 24 chemicals that are known to be used in UOG activities for 5 different types of hormone disruption. We measured receptor activation and receptor inhibition with our reporter gene assay. So these are shown in slide 24, the 24 fracking chemicals that we tested. And I'll come back to these in a minute. You obviously have them and can study them at your convenience. So what I'm going to show you then is just an overview of what we found. I have organized this slide according to the chemicals that were the most potent. So in slide 25, we show estrogen receptor inhibition. And all the slides on activity, actually, I'm going to show you are inhibition of activity. They are not stimulation of activity. So we saw the majority of chemicals had some ability to disrupt estrogen receptor signaling. And then over on the far right, we see a mixture, for example, of 23 of those chemicals, diluted tenfold relative to the majority of the chemicals to the left, can activate estrogen receptors maximally. Androgen receptor inhibition, we actually saw a very similar profile as far as the number of chemicals that acted to disrupt androgen receptor signaling. Progesterone receptor inhibition, we saw fewer chemicals, but a few that were maximally active. Glucocorticoid receptor inhibition followed with slightly reduced activity, and then the fewest that we saw were thyroid receptor inhibitors. So summarized in slide 30 is the activity of the 24 chemicals on the 5 receptors that we tested. And what you can see is we saw a handful, or not even, just a couple of chemicals that activated estrogen, progesterone, or thyroid receptor. But we saw a huge number of chemicals that inhibited these receptors. So, of course, we're not exposed to single chemicals alone. So what Chris Casotis, a very talented graduate student in my lab, did was make a mixture of 23 of these chemicals shown in slide 31. And he tested that mixture for its biological activity in vitro, which is shown in slide 31. And so what you can see is just a varied amount of endocrine-disrupting activity for those mixtures, but an increased activity relative to the single chemicals. So what he has gone on to do is to test this mixture of chemicals in vivo, and that's just an outline shown in slide 32. So we will be reporting those results in the next month or two, so I will wait to update you on what he has found there. Additionally, we have conducted a large field study in Garfield County where we collected 48 sites. And then in slide 34, it shows you just the approach that we will take to look at what chemicals are responsible for that endocrine-disrupting activity using an effects-directed analysis. And just by way of a teaser, in slide 35, I have highlighted in yellow the chemicals that we have measured analytically. Dr. Chung-Ho Lin and Don Tillett, our collaborators, have measured these chemicals directly in some of the wastewater that we have provided with them. And we know that these chemicals are endocrine-disrupting chemicals. So finally, in conclusion, hormones and EDCs work at low concentrations. EDCs are associated with disease in people. We found that 23 of 24 chemicals tested showed some type of EDC activity. And surface and groundwater from these sites had greater endocrine-disrupting activity than control sites. So in slide 37 is our research team, and I will end on slide 38 with thank you.
Thank you very much. That's a lot of information, and I really appreciate the presentation. So let's open up to questions from the blog now. Erica, do you have questions for us now that you can bring forward?
I do. I have a couple of them here already, and I'll continue to refresh as others are typing theirs in. So I'll start with one from Ann Bristow. she would like to know, how do you respond to industry critique that the Colorado data, CO data, don't control for land use EDCs, pesticides and herbicides, for example, with the West Virginia well site, since that looked forested?
Okay, so let me see if I can repeat the question. Criticism that we did not control for land use. Absolutely, we took that into account. We had about 40 samples from 7 or 8 sites in Garfield County, so it was impossible to control. We didn't have enough power to control for that in a systematic way. However, with that said, we did control for it in the sense of none of those sites were used for agricultural operations, either plants or animals, in the recent past. And then West Virginia is its own control, because we were measuring one creek in the exact same area and comparing above, on, and below this injection well.
Okay, thank you. From Jacob Matz, can you say a bit about the relationship between EDCs and air pollution surrounding unconventional oil and gas sites? Is it possible that these chemicals could become airborne throughout the process of oil and gas production?
Yes, absolutely they do. Many of these chemicals are volatile organic pollutants, and they are either semi-volatile or volatile. We have not done. Others have looked at these chemicals in air and found them. The majority of these chemicals they have found in air, certainly not all of them. For example, naphthalene is one that has been found in air and in water, and is quite a good endocrine disruptor in our hands. We actually would like to, and are kind of in a methods development approach right now, to capture from air and concentrate air, capture the chemicals, elute them, and test those for endocrine disrupting activity. So yes, we think it's absolutely possible and probable.
Thank you. And I know Che did a call looking at some air pollution related to fracking sites a while ago, so I'll try to remember to put that up as a link as a response to that question as well, if anyone's interested. From Lizette Van Vliet of the Health and Environment Alliance in Brussels, our colleagues in Brussels, she asked, can you please comment on how geographically and chronologically widespread the health, environmental, social, and other impacts of EDCs in oil and gas extraction and exploration can be? So geographically and chronologically widespread, how far does the reach go?
Wow. Yeah, that's a pretty big question. Chronologically, as I said, our initial study is relatively small. However, with that said, the spills that had happened from 2 months before to over 5 years before. So it is very possible that those, a lot of what we know about these chemicals is they associate with dissolved solids and sediment, and they can then be released over time back into the water. So chronologically, certainly I think that we are just beginning to understand the long-term impacts, but I believe they could absolutely be there. Geographically, that's ongoing. Certainly, from the little bit of epidemiological data that, for example, has come from McKinsey and Atgate, they looked at developmental effects and looked at birth defects in babies whose mothers lived in proximity to operations, and they used 10 miles as a cutoff and found increased incidence of neural tube and heart defects in children whose mothers lived within 10 miles of drilling, and that relationship did increase the closer you lived, the closer mom lived. So that's at least a ballpark for a geographical reach.
Okay, thank you. Joe Rochester asks, why do you think most of the activity is inhibitory, and do you think the mechanisms are strictly receptor blocking?
Yeah, great question. So that is the short answer is that is the next phase of this work is to look at the mechanism of action. It is very possible that it is the inhibitory effects are upstream or downstream of the receptor, particularly, actually I'll just stop there by saying that is our next phase of research, to look at whether it's a direct binding or indirect.
Okay. And Kembra Hodeshel, I apologize if I'm mispronouncing that, but how do you formulate your mixture doses? How do you determine how much of each chemical to add to your mixture?
Hi, Kembra. So what we did in this case, since there are many approaches and we had to pick one, was we chose an equi-mass concentration. So for the in vivo, that was for the in vivo work, so in animals. In tissue culture, where we have much, much more latitude, we've made all sorts of combinations. So we've used equi-mass, equimolar, and equipotent. So then we've chosen receptor-specific mixtures and tested those for additivity, synergy, and antagonism. And those will actually be forthcoming in Chris's next paper, that approach. So with that said, we don't have that flexibility in animals since it's so expensive to do that. And their approach there was just to use an equi-mass mixture for those chemicals. In that case, as far as choosing the concentrations, we really tried to bracket the concentrations that would be found in, say, very concentrated produced water, but then also downstream surface water.
Okay, and Cheryl, I'm just going to ask one final one, and then maybe if you have one, and then we can close out the call. But Dorothy Fowler had sent this question to me actually a couple of weeks ago prior to the call, saying that Ohio is now spreading or can spread brine from fracking flowback on about 170 of their cities, townships, roads to melt snow and ice. And so she just wondered if you could speak to the potential health effects of this process.
I think that is a risky proposition. So spraying those on roads, as of course other states have allowed as well, allows for runoff into surface water. And, our water treatment plants are not designed to remove these types of chemicals. Some of them are removed and some of them aren't. So I think it really increases the likelihood of water contamination by doing that. Okay, and Cheryl, did you have a question that you wanted to ask?
Sure, we don't have a whole lot of time, but one quick question. Susan's excellent presentation, kind of a long-term question is, some of the chemical effects that you subsequently explored had to do with reproductive health outcomes, but I'm wondering in the long term, would you expect to see rising rates possibly of cancers in some of these locations where contaminated water is being used for drinking water? And I'm just concerned because many of the chemicals you've been talking about are the same kinds of chemicals that firefighters may be exposed to at different levels when they fight fires, particularly the BTEX chemicals, benzene, and so on. And we didn't talk about pHs as well as pHs. Could you hazard a guess?
Yeah, absolutely. We would predict, and some folks are starting to look epidemiologically for the health impacts of exposure to these chemicals, particularly developmental exposure, but also long-term chronic exposure. And we would expect potential impacts on cancer rates, but particularly on fertility. So we know from the literature, from animal studies, from occupational exposures to a lot of these chemicals, we see effects on fertility, both reduction in sperm count and also the ability to get and stay pregnant.
Okay, thank you very much. I think there are probably a lot more questions that we don't have time for, but Susan, if you're all okay with this, I would like to suggest that people send us additional questions, and we might send them on to you, and you can respond to those people who are on the call if you have time to do that, so we can sneak in a few other questions after we've had to close down the call. Would that be okay with you? Great. Okay, well, I want to thank you very much for talking with us today. It's extremely important information, and you're doing great research, so thank you again.
Thank you. Bye.
Okay. Okay, Erica, can you close the call?
I can, thank you. I apologize for that. I'm not quite sure what that was. An MP3 recording of today's call will be available on the CHE website soon. We have 2 calls scheduled for next week if you're able to join us. The first will take place on Wednesday, February 25th, and is titled Placental Toxicants and Disruption to Development. And the second will take place on Thursday, February 26th, and is titled Environmental Influences on the Thyroid System. And you can RSVP for both of those from our website. I will be sending out a brief survey following our call today, and we hope you'll take just a few minutes to provide us with your feedback. It will help us to assist in planning for future CHE calls. And with that, I just want to thank Dr. Nagel one more time and Cheryl for her moderation, and I wish all of our participants a very healthy day. Thank you so much.
Thank you.
Thank you. Bye
Thanks
now.
a lot. Okay. Bye-bye.